Hello, everyone. Welcome to today's live broadcast, Accelerate Bioprocess Development Using the Modular Automated Sampling Technology, or MAST, platform. This is Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be your moderator for today's event. We're pleased to bring you this web seminar presented by Pharmaceutical Technology and sponsored by Capsugel. Capsugel designs, develops, and manufactures a wide range of innovative dosage forms for the biopharmaceutical and consumer health and nutrition industries. The company's unique combination of science, engineering, formulation, and capsule expertise enables its customers to optimize the bioavailability, targeted delivery, and overall performance of their products. Capsugel partners with more than 4,000 customers in over 100 countries, create novel, high-quality, and customized solutions that align with its customers' evolving needs and benefit patients and consumers. For more information, visit www.capsugel.com. I have a few important announcements before we begin. This webcast is designed to be interactive. We encourage you to ask questions during the event. You can submit your question by typing it in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. The speaker for today's webcast, Dr. Clint Pepper, has spent more than 20 years in the biologics, pharmaceutical, and medical device industries, creating products, developing processes, and manufacturing biopharm compounds in development, clinical, and commercial environments. Clint's helping Capsugel create the Modular Automated Sampling Technology, or MAST, automated sampling solution that can be used in any application where maintaining sterility of the manufacturing process is the highest priority. Thank you for joining me today, Clint, and I'm going to turn the presentation over to you.
Hi, everyone. We appreciate everyone taking time this morning to come out and listen to a discussion about the MAST technology. So today I'm going to talk to you a little bit about how the MAST technology is being used to accelerate bioprocess development. And so I'll kick it off. First, I have an agenda, what we're going to talk about. I'm going to go through an introduction to the MAST system and why you would want to do auto sampling. Then I'll go over how the MAST modules are configured so you can understand the overall MAST system. And I'll get to the heart of the presentation, which are 3 case studies. One talks about the integration with the Nova BioProfile FLEX. The second one talks about how you can facilitate novel development approaches in your lab. And the third was how you can integrate product quality attribute control into your systems. Following that, I'll cover some points on the MAST deployment and reliability, recent development efforts, and so, we as an engineering firm go out to end users all the time, and what we've been hearing is that people want to consistently achieve their target quality attributes. And we believe that the product quality attributes are a function of 2 things. One is the bioreactor environment. So this is your pH, temperature, the solution adding, things like that. But we've also been learning that it's also a function of the cell state. This example from Journal of Biotechnology from Lee, or the bifurcation of lactate production in cells. You can't see it early on in the culture because the cells are in a different state. And so understanding both how the bioreactor is running and how the cells are performing is critical to achieving your product quality attributes. And to understand these, you need 2 things to happen. Right now, given the current technology, to get this data, you have to take samples. And if you want these things to be controlled, really hit your quality target attributes, you have to do this automatically. But taking the samples is only part of the game, because if just taking the sample doesn't give you anything unless you can automatically analyze it. So, as an overall discussion of what MAST is, what if you could automatically collect whole broth and cell-free samples? This is something that is very challenging in the industry for a very long time, is making sure you retain sterility of your bioreactor and get samples that are consistent. As an engineering firm, we were asked by some end users to design these auto samplers, and we invented the Sample Pilot SP100, which is used for stainless steel bioreactors that are sterilized in place, and the Sample Pilot SP200, which is designed for development scale bioreactors. And these are designed specifically to maintain bioreactor sterility samples. So there you can take these whole broth samples, but we also designed a cell removal system which allows you to remove the cells from samples for different types of analysis. So if you could take these types of samples and then automatically analyze them for cell density, viability, using like the high-res or the Nova, or get metabolite or other information like Nova, the BioHT, and other devices. So imagine if you could collect this type of data and then also take the cell-free samples, do some processing on them, like do protein A or digestion or dilution, so that you could get the sample just the way you want it, and then do an analysis on a patrol or other type of UPLC or mass spec. You could do all of those things. Then we believe that control of your product quality attributes is possible. So if you look at a typical bioreactor setup, you have a bioreactor sample gets pulled into the Sample Pilot. Whole broth samples get analyzed by your bioprocess analyzers. Your sample goes to your cell removal system, and there your cell-free sample can get processed and analyzed using HPLC. Then you're able to pull all this data together. You have in the green and the yellow. That's gray. In the gray there, you have your online data, capacitance, DO, and then you have all your at-line data, which is all the stuff from your cell analyzers and your HPLC. Once you have all that data, now you can begin to create predictive models, which then you write your control algorithms, and ultimately tune your process levers to control your nutrient productive feed, like that. Now you're really able to get product quality attribute control. So, this talk is going to focus on like I said, increasing the value of processes using automated sampling. And the basic level, and these case studies I'll talk about, show that the value of auto sampling itself, just making sure that everyone who's working in your lab is doing high-value tasks, not just collecting sample analysis. In the second case study, I'll show you how in our labs we were able to come up with some novel ways of doing the process development process that dramatically accelerate both the development and the scale-up. And then the final case study talks about how you overall implement the product quality attribute process. So one of the things that I want to talk about right now is why is auto sampling so difficult? And years there wasn't a good. And that was for three reasons. One, maintaining sterility was always an issue with auto samplers. Then also reliably getting the sample to the destination was always a challenge. And then because of viscosity issue, the high cell density processing. And then the clogging of, over time, cell debris accumulation eventually clog these systems. So we designed the Sample Pilot to resolve these challenges. So the first one was we increased the sterility insurance profile. So the sample pilots are autoclave sterilized and as part of the preparation process for the bioreactors. And then any fluid flow into the sample pilot, the desannitant or purging gases that we all flow through sterilized 0.2. Now the key to the sample pilot is this: is that there's a pump built directly in the body body of the sample pilot. What that allows you to do is, after you have, you're ready for a sample, you have an aseptic area draw in the sample. So there are isolation valves. There's a bioreactor isolation valve that's interlocked to prevent it opening at appropriate times. And when the vacuum pump is drawing, that's inside the sample pilot, it draws the sample into this aseptic zone, which has been shown over thousands of samples that end user to resolve sterility issue. Now, once the sample is out of the sample pilot, through these filters, we push a sanitant, like 70% IPA or ethanol, which can flush through the system and be held for certain hold times for sanitization of the system. And then we follow that with a purge gas to blow that sanitant out and get rid of it. The next part was reliably getting the sample there. So we push the sample, not pull the sample. Other auto samplers attempt to draw the sample using pump or that type of at the destination. The problem with that is you can only draw a sample using pounds of suction. But instead, we push the sample using this positive displacement pilot, and we can push at pressures exceeding 70 pounds. For a particular client, we push using an SP100. We push a microbial sample that's yogurt-like, about 80 feet to its destination. In this system, we have this, again, a static seal positive displacement pump, and the valve that isolates this pump from the bioreactor is interlocked so that it cannot open down to the fundamentals of the programming. It cannot open unless certain conditions are true, and one of them is if the pump is ever running, that valve. And then one of the other things, a new feature we just added was that you can push the sample with purge gas if you're trying to minimize the volume that's being. You can draw a sample and then use a gas to push the sample volume you can. Lastly, on this, we clean and sanitize after every sample. This to reduce the clogging issue. So all sample contact components are flushed with sanitin after every sample, and then the samples and the sample lines are flushed all the way out to the destination. So we operate the pump during this process. We flush the sanitin out, comes through this sterile barrier filter, and then we allow these units to sit for specified hold times. The valves inside are specially designed to allow very smooth flow without edges or creases where cells can accumulate. So there's a lot of work around that to reduce clogging, which is a growing issue. So let's talk now about how the MAST modules are configured. The system is the M in MAST stands for modular. It can take up to 10 sample sources and send those up to 4 analytical destinations. So this first is the basic type of MAST system you can have. You have a computer interface, a GUI that has your scheduler, and we don't monitor the system. Then we have controllers that drive all the pneumatics to operate the sample pilots. This setup will run one sample pilot and send it to one destination, like this Gilson liquid handler we for Nova BioProfile FLEX, other items. The idea is that you can start simply using one bioreactor sample source, but then it can be easily expanded. So in here, we add this item called the Sample Navigator controller. We add that. You add enough outputs to run 5 bioreactors, and the Sample Navigator box, which is like our traffic cop, it routes the samples to. And this can be readily expanded up to 10. So in this diagram here, you'll see that that by adding 2 Sample Navigator controllers, you can run up to 10 bioreactors and send those to an item like this Gilson or like this Nova or other device. Now, once we have routed the sample to a destination, then you say, well, what happens if we want to send it to more than one destination? So then we add this other control box called this Analytical Navigator. which has header valves which will direct the sample to one of multiple devices. So let me get into the case studies here. There's images of SP100, which is mounted on a microbial bioreactor, or SP200 mounted on a bench scale bioreactor. How they are used in real life. So in this first case study, we're going to be talking about integration with the Nova BioProfile FLEX. The goal here was to reduce the non-value-added work by providing samples directly to the FLEX without human intervention. So we know what time or next time scientists and engineers drawing samples, handling the data. So what the approach here was that we have an externally mounted sample cell that's mounted out of the way, allowing manual samples, but you can see how it's moved into place for automated sampling. And our system bypasses the Nova RVM and SPM. When setting up the sample in the MAST system, you schedule it and tell the MAST system the parameters, because it has a bunch of different tests you can run. So in this first case study, we pulled data from 5 different cell culture batches, and these were done under a wide range of conditions. Some of the samples were collected with SP100 units. We had vessel size ranging from 1 liter to 500 liters. Some of the batches were performed by end users, or some are here at Capsugel. Cell culture processes range from regulatory tox lots to small-scale development experiments, and samples were drawn at different facilities, bioprofile. And the main goal here was to compare the manually collected samples with mass samples, and so each data point, both a mass sample and a manual sample, was collected, and we created parity plots of the cell density, viability, the other attributes. But overall, what we showed was that the mass samples were representative of the manual samples, with variability documented. So this first slide shows the slide on the left shows the total cell density, viable cell density, and viability normalized in a parity plot, and you can see that within the error, you know that very good correlation. Same is true when you look at the pH parity plot. Very nice correlation. Looking at the other FLEX data, you have salts, sodium, ammonium, and potassium. Curve, you can see excellent correlation, and the Nova has a higher accuracy measure. And then the metabolites, glucose, glutamine, glutamate, and lactate all had also a very good correlation. In addition, the dissolved gases, the PCO2 parity plot shows a good correlation. And while the CO2 looks like it's not as well correlated for the fact that you're taking a sample where cells are eating the oxygen in the sample as it's processing. This is actually positive data. So we're very happy with these results, and we think that this shows that the Nova automated sampling can replace manual. System up like this will save your operators and techs and scientists a lot of time today to do more value-added tasks. Second case study this work we did in our lab is a measurement of galactosylation and perfusion systems, step changes in galactose. Here at Capsugel, we used a perfusion setup to collect data on dynamic cell response. Remember in the beginning of the presentation I talked about cell state. So this is getting to how cells respond and react to understanding the dynamics of the. That's critical if you're going to product quality attributes. Here we sampled whole cell broth. So you see on this picture, there's two bioreactors here, both running at perfusion. And in the upper red circle, you'll see the sample pilot that's connected to the bioreactor, and that's pulling cell broth from automated analysis by the Nova. And then down in the lower red circle, you see that we also have another sample pilot on the permeate outlet. And there we're taking that samples to a Gilson GX1 liquid handler. And from there we did the glycan analysis. All of the data from the collected by our internal database automatically processed. And we were able to sample at very high rates when we were studying these. So in this case study, what we did was we looked at a step response. We subjected the perfusion reactor to step changes in galactose concentration. Those are the lines there. And then we allowed the galactose oscillation to reach a new steady state value for the time outputs. In this, we used a perfusion system that ran at constant cell density, constant feed rate, and constant volume. And this is the data we got out of that. And first, that you can see that the galactose is consumed in response to the step changes. So what we had is, before day 19 there, you see that the cells, we didn't add any galactose, but the cells are actually producing some galactose. But then as we started to make the step changes, you see on the orange dotted line is the feed concentration, is the measured concentration. And then the output, you look at the galactose oscillation, until the galactose reached about 100 micromolar, there wasn't much of a response. And then after that, the galactose oscillation started to match with the galactose. And the critical part about this, look at the dynamics of what's happening here. So first for the galactose, what you see is we made a bolus of galactose and then changed the feed, so we should have had a nice sharp peak. Instead, you see that the actual galactose is down a little bit, and that's saying that the cells are increasing their galactose consumption. And that's a dynamic response. And then looking at the galactose oscillation, what you're seeing there is seeing that it takes time for the cells to come to their new steady state. So what you get is you get the rate of increase of the galactose oscillation response, and then the new steady state value. And if you were going to do any kind of a feed-forward control mechanism, this cell response rate, the time it takes the cells to change after getting this galactose feed, critical parameter in the whole scheme. The idea here is that we're looking at how to do development more novel ways. Being able to take automated analysis allowed for this new approach where for every 3 or 4 days we can change a parameter and see a result instead of that batch process. Make lots of changes, run something, dramatically reducing your overall development time. And this is just one example of a way that you can change your entire approach to how you do development by having this automated system. At the end part here is that one of the things that is really critical about this is being able to take more samples when you need them. So if you want to watch this dynamic response, you'll see that without, if you're taking samples once a day, you're not going to see this dynamic response. But if the cells you're expecting the cells on Saturday night at 1 o'clock in the morning, you can go in the lab, or you can have the MAST system take samples every 2 hours for you. I think that's the critical part about how auto sampling is different. Adds that ability to capture that dynamic. And that third case study talks about how we automated UPLC analysis for cell-free mass samples with pre-processing of protein A acid preparation. So kind of we're going to also feed this through about how the MAST system works a little bit. The MAST platform talks to the cell removal system, the Gilson, and the waters to make sure everybody's ready for samples. Otherwise, the samples aren't initiated. In this particular application, there were 2 SP100 units drawing samples that were routed through our sample navigator and then into our cell removal system, which I will talk a little bit more about in a little bit in the development section. So the cells are removed from the sample and then put into our Gilson liquid handler. This is we partner with Gilson, and they've been a very good partner for us and very helpful. And we've been able to lots of different types of processing. In this particular example, we did a protein A preparation or an amino acid preparation samples. Then the samples are transferred from the Gilson to the UPLC, where the MAST system is able to run methods called out when you schedule a sample. So our system can talk to the UPLC and fetch back all the methods that are available in the UPLC. That way, when you schedule the sample, you can load up that method. Sample gets dropped off, it will run the correct method for your sample. And so one of the things that kind of shows some consistency to the Gilson, we wanted to verify that the protein A method was consistent between more other typical systems that are used. So we did a study where we took 12 replicate samples to demonstrate consistency. 6 of these samples were run on the traditional protein A process on the didactics, and 6 were run on the automated protein process by the MAST system using Gilson. You could see that the resulting protein recovery was very consistent. So moving on to some other, to discuss the MAST deployment and reliability, where we are now. I mean, we launched this product in October. We've been developing it for about 4 or 5 years with multiple clients. So 10 companies have used the MAST system out there. A lot of them were prototypes, and we now utilize the system, and but there are 10 MAST systems out currently. 10 companies and 11 facilities that are using. We've deployed 44 of the SP200s and 7 of the SP100s. We have 5 cell removal systems out in the field and 7 of the Gilson. During that time, we've taken thousands, if I could really count, maybe tens of thousands of samples taken by these engineers. Not had a single sterility failure that's been linked. That's really demonstrated our ability to bioreactors. We've used this successfully in cell culture, microbial, and downstream applications. Several places, samples after protein A step or various other steps in the process. And if you ever come out to our labs out here in Bend, Oregon, you will tour our lab and see how we're always running systems, tens of thousands of cycles of on-robustness testing. Now I want to talk to you a little bit about the recent product development efforts that we have. And so we're going to go through some of the next steps or next things you're going to, some of these things that are just are coming out with. First is our commercial controllers. If you've seen other systems out there, they could do 8 sample pilots or sample sources, and this new system can operate 10. It works with both SP100 and SP200 units. It has new features in it to better regulate flow of gases that push samples around, so we can use high and low pressure to. We've added a new liquid sense technology into this system, which can detect whether you expect there to be sample or not. So if sample's there and it's shouldn't be, you get an alarm, or vice versa. Yeah, some of the bigger things here is we also incorporated lessons learned from all the prototype units we had. Several of our end users had these prototype units, a lot from them. Improve the components, design the system to be more software, and these units are completely modular and stackable, and back from installation to very, very easy to install as far as fit and make. The commercial SP200 units, maybe they came out last year. You can see definitely an improvement over the original design. All the pneumatic connections are all now internalized. I'm using more standard filters for flowing the purge gas and the sands in through. Yeah, smaller is a little smaller, and all the connections are on the top, but they're a lot easier to get to on the bioreactor. We have improved and simplified the valve seat design, which and some other things we design areas. And it's and we have a fully integrated pneumatic umbilical. So now that right, you had to connect. Now it's a lot more straightforward. And we, just down in, like I said, in installation, you can install one of these on your bioreactor. So there's, it's pretty fast. Wanted to kind of also show how you connect a SP200 to a single-use bioreactor. We have two ways that do it. One is using all clean pack adapter, which is shown here. Just like when you connect a probe or a DO probe to a sub, you can utilize this adapter. Then we also can connect during sterile well too. And this setup you see right here is actually going to be a bioprocessor now. Come by and bioreactor. Now I'll talk to you a little bit about our cell removal system. This is another thing that was some of our end users asked us to design and develop, because this we have with the see the auto sampler. The cell removal system left, and the right is the cassette that goes to the cell removal system. And this is basically how the system works. We use a hollow fiber technology where the sample is brought into one of the syringes and then pushed back and forth through the hollow fiber, collecting healthy permeate. And this is designed specifically for many reasons. One is that cell removal process is conducted completely outside of the bioreactor barrier. And this was an important requirement for our end user. Because the filtration cassette can be changed any time without impacting the bioreactor sterility. So if your filter gets clogged, or you start to get gel layer formation up on the filter, and you're starting to impact your recovery, those types of things, you can just change the cassette. You don't have to worry about filters in the bioreactor itself, and it's possible to do. Also, it's not a sterile barrier. So if any kind of clinical GMP type operation, a membrane that's inside or acting as a sterile barrier of your bioreactor, go through sterility validation, and because of this, you'll. And we focus on the hollow fiber strategy because using tangential flow is based on fusion, very high cell density. They're using better than cells from permeate. Cell removal system can consistently separate cells from permeate for up to 30 samples per set. after the system is full flexibility with respect to sample processing, and after every sample's processed, we run a flushing and cleaning process to. One of the issues that we are working on with many end users is that the protein recovery is dependent on the cell line or the protein that needs to be processed. So this is where we've been working with different suppliers of membranes to right surface area and the membrane type, but also the right processing shear rates across all the fibers. So this is still a development process that seen some good results. Depending on your protein, your cell line, there will probably be some development associated with the exact performance you're looking at. Due to the nature of hollow fiber processing, protein recovery will tend to diminish over time. That's why you'll so we'll continue to develop this, and this is something that we're excited about optimizing.
Clint, this is Rita. I'm not hearing you right now. Can you check your microphone, please?
Can you guys hear me now? Is that okay?
Yeah, you're a little distant. Maybe if you can get a little closer to the microphone.
How about that? Is that okay?
That sounds great. Thank you.
Okay. All right. So also we have been working with Roche on working with integration to the CDEX high-res. We are working with one end user to integrate the high-res with our system. We've established communication protocols with the system. We've developed the ability to initiate the samples. Because the high-res measures up to 10 million cells per mil, we're designing a in-process dilution system to do up to a 10-fold dilution, which would allow measurement up to 100 million cells per mil automatically. And we're going to be deploying this in the second quarter of 2017. Similarly, we've also working on with the same client a integration of the BioHT, and that communication protocol has also been established. And we've demonstrated the ability to sample BioHT via a remote trigger. And again, we're going to be delivering this prototype solution in the second quarter. Some other things to think about, this if you're running this in your pilot plant or development and you want to take this process into GMP, we wanted to let you know that this system was specifically designed to be able to be taken into the GMP environment. We've been using the industry-accepted hardware like Allen Bradley and Ignition solutions. We have designed everything to the industry standards with traceable components and complete bill of materials. The product contact materials that your bioreactor sees are either PEEK, which is a very low extractables plastic, one of the best plastics out there, or a 316L stainless. And then the sample contact has some more components, but all regularly used in sample contact environments. We can put validation packages together upon request, and we can bolt on software modules to meet your 21 CFR Part 11 requirements if needed. So you could take this into your clinical or your GMP applications, especially if you're doing something like product attribute control. You have to be able to utilize these solutions all the way through from development into commercial. So in summary, talk about how we accelerate development. The automated sampling and automated analysis tools now allow the development of biotechnology manufacturing processes with real product quality attribute control capability. As we showed you in the first case study, For routine assays, you can free up resources to work on higher priority projects. The second case study, the auto sampling allows creation of novel ways of creating, developing new development approaches like the perfusion cell setup we described, but that's just one example. And knowing that you can auto sample and auto analyze gives you a whole new way of attacking development. And then last, we showed in case study 3 how by taking automated sampling and analysis paves the way for integrated product quality attribute control. So summarizing the advantages of the overall MAST system, you can create an aseptic zone to draw the sample in, significantly increases the sterility assurance profile of the sampling operation. Having the positive displacement pump on board the sample pilot has demonstrated to be very reliable to get samples to their destinations, even if it's extended distances. On the cell removal system, it's important that the cell removal component is external to the bioreactor for the sterility assurance components and also for being able to change it without interrupting your bioreactor operation. Is very important. Our overall system is a closed system, so we can utilize high pressure for the cleaning and sanitizing and drying processes, which is important for reducing clogging and getting your system to be reliably and consistently back to its original state after every sample. The whole system is completely modular, so you can start very simple and add modules as you need to. The bioreactor time is quick, and you can take this technology and scale it up through GMP manufacturing. That's my presentation, and I really appreciate everybody's time, and I look forward to your questions, and or you can also call me, go to our website, and we have some numbers there to contact us. But look forward to talking to you, and again, thank you for your time. It's been great.
Thank you very much, Clint, for that presentation. Before we take questions, just like to ask the audience to participate in a brief polling question, and you can click directly on your screen to submit your answer. So the question is, would you like Capsugel to follow up with you after the webinar? Yes or no? Again, the question is, would you like Capsugel to follow up with you after the webinar? Yes or no? And a few more seconds for everyone to submit. All right, and thank you for participating. So let's move on to the question and answer portion. Just a reminder to the audience, you can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. All right, so here is our first question. How much in milliliters is consumed with each sampling?
Oh, good question. So the sample pilot SP200 draws samples in this pump in 5 milliliter increments. And so generally the first sample contains the residual in the dip tube and in anything that may be in the pump. So the first 5 mils is usually sent to waste. And then it depends, you pull a sample in 5 mil increments. So if you're not concerned about volume of sample consumed like a big reactor, we generally push liquid with more liquid, so you fill the line very reliably delivering sample. If you are concerned about sample, then you're probably going to pull another 5 or 10 mils and use our air push system to push the sample to the destination. So the answer is that if you're really concerned about volume, you're talking about maybe 15, 20 mils of sample you're going to consume per sample. And it can be higher than that. The SP100 pulls much larger samples, but it's designed for larger bioreactors. It pulls 50 mil samples at a time.
How do you deliver samples to UPLC?
So right now what we do is we send, like in the presentation, I don't know, can I go back to one of the slides? Is that possible?
Sure. Do you know which slide it is? You just click on that slide, and then you can push that to the audience.
So in this item here shows that what we do is we send the samples through the cell removal to the Gilson, and then from the Gilson we interface with we fill up a sample loop on a 6-port 2-position valve, and once we put the sample that's in that loop, we switch it over to the waters, and then MAST communicates that the sample's there, and the waters draws that sample in. That's our typical process for doing that.
Okay, thank you. Next question. Does connecting the auto sample loop to an analytical instrument such as the Nova FLEX limit or make it more difficult to use?
Well, the connection of the Nova is relatively straightforward. We've done that quite a few times now, and so that process has been pretty well established. New connections sometimes take some time to develop and make sure we work through prototypes and get samples there, more reliably. But it's connecting the Nova makes it very easy to use because you don't have to draw any samples or anything. And our system is set up so that once you connect the sample pilots to the bioreactors and connect them to the system, everything else is already pre-connected. So it's actually relatively straightforward.
So here's another question. It says cell removal system 30 samples per day or per system. Then how do you prevent samples from contaminating each other?
So the cell removal system can take 30 samples, and if you have a bunch of different bioreactors, pulling samples, and that's those are just samples that get processed by the system. So if you have multiple bioreactors, they're all feeding into the cell removal system. In between each sample, we run we blow the cartridges in the cassette down with air, and then we run a flush solution through the filter and through all the tubing, and then blow that down again before the next sample. So there's a cleaning or flushing process that occurs between every sample. There's 2 solutions that are supplied into the cleaning system, so you can also apply cleaning solution. If you're concerned about crossover, you can do a more rigorous cleaning and flushing, and that consumes more time. So, finding out exactly what your application is, we'd be happy to talk to you about, fitting, meeting your needs or that. Our system is completely flexible in how you set up the sequence. So, some people may not need such a rigorous flushing, and therefore they can process more samples. So there's a lot of flexibility in the system, and so we're eager to talk to anybody about it and find out what their exact application is and making sure the system meets their needs.
All right, thank you. Could you clarify the point about protein recovery by the cell removal system? Does this mean titer cannot be accurately quantified from cell-free samples?
So like with any, just as like with perfusion, you're always having some kind of a membrane loss, protein loss across the membrane. Generally, your recovery is, 90, 95%. But as over time, as gel layer forms on, say, a perfusion system, you'll see is that the protein coming out of the perfusion system is not as consistent with taking a whole broth sample and spinning it down by centrifuge. And so what we are kind of using that same model in understanding how protein recovery is in our hollow fiber system. So you're always going to have some loss of protein. And so what we're striving for is to be in the upper 90% recovery range. But we're still working through that, and we find that some proteins and some cell lines have more issues than others. So it's also finding the right membrane type and the right processing conditions for your particular application. And that's, we generally work through that on a case-by-case basis with people.
All right, thank you. Next question, can you comment on the rate of failures for these instruments?
Well, we have a very good history. We had several end users. Like I said, we've taken thousands, if not tens of thousands of samples without a sterility issue. The new commercial enclosures took a lot of the issues that we saw over in the early part of the development and integrated those solutions in both from a software perspective and a hardware perspective. So we're seeing some very good, reliable results now, and we continue to work on this product. We're committing to make it a very high quality, very reliable product.
All right, thank you. Here's a clarification of an earlier question. When the MAST is connected to a Nova FLEX or a CEDX etc., does it make it more difficult to manually run samples through those systems when you have the sample sets to process that are not part of the MAST setup?
Oh, okay. So both of those systems still allow you to do walk-up samples when you're doing your automated samples. In the BioHT, you could always put a new sample rack in next to where our apparatus is, and the same thing is true with the high res, where you can still come up and schedule samples. And I think it's just a matter of coordinating between the manual and the automated samples. Our systems look to in the high res and the Nova. If we see that the Nova is busy, then we will queue our samples. Our software system has the ability to wait or queue samples. So if one of the devices is in use for something else, it'll wait its turn to draw that sample. And we don't draw the sample until the devices are ready. So you're not having a sample sitting around, waiting for the sample to be taken. So the Nova and the BioHT and high-res all have the ability to walk up and interact with the system independent of what the MAST system is doing.
Okay, great. Next question, how much IPA is needed for cleaning, and how is the used IPA disposed post-cleaning? So
depends on your particular process. We generally consume per sample about 100 milliliters of IPA, maybe it's like 80 milliliters. Depends on how much you want to flush through, how long your lines are, some other components. We have a Sanitant supply station that holds about 5 gallons of Sanitant, and then that all comes back through to our waste system collection, which is generally a carboy. And we've had different solutions. Sometimes we provide those in smaller containers so that if you have EH&S issues with IPA accumulation, some people have put the entire apparatuses inside a flammable cabinet. So I know that there's some issues around using that, but we've got some solutions for that.
Okay, thank you. Do you support other chromatography platforms other than the Waters Patrol system?
So the Waters Patrol system is the ones where we have completed, successfully integrated. We also have several projects that are starting with other different UPLCs. And it's really all it's about is about the interface to the software. We feel that getting the sample there physically is not going to be as challenging as it is, communicating. So as long as the company that if it's either uses a ChemStation or Chromeleon or whatever those other software programs. Currently, we communicate through Empower to Waters. So if you're using Empower to run your, say, a different brand of UPLC, then that would be pretty straightforward because we have developed that communication protocol. But connecting to to a new device through a Chromeleon or something would have to establish that. So there's usually a development program where we would go through and work with the software developers for that software company to complete that integration.
Okay, one final question. Do you have a solution, or are you working on a solution to pair with the Amber platform?
So one of the things with Amber is that that's a very small volume system. So because of their 15 milliliters in size, we find that our system draws too much volume for that. They do have 250 mil reactors, and so we currently don't have a program because we think those might be too small for the sample sizes we draw. We do have a program right now ongoing that's in early development to create a much smaller sample pilot, but that's still a ways out. But we could probably use that with the 250 Amber, but right now because that's in early development, I'm not really focused on that yet.
All right. Well, thank you very much, Clint, for your presentation and for your answers. I'd like to thank the audience for participating and thank our sponsor, Capsugel, for making today's webcast possible. This program will be available for on-demand viewing through February of 2018. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. So I invite you to forward that announcement to any colleagues who may have missed today's live event. Also, we're going to put a sponsor survey out once this webinar is over, so please take a few moments to complete that. Thank you again for joining us, and we'll see you next time.